You are a member of an alien civilization in the microcosmos. You have tiny spaceships the size of neurochemical molecules that enable you to travel inside the human body. Today, your teacher is taking your class on a field trip to explore the human brain. All around you, you see a vast network of neurons connected with each other in a complex and thread-like structure. The neurons do not exist in a vacuum, however, but are surrounded by glial cells of similar sizes. Glial cells hold neurons in place and insulate them from another, to supply them with nutrients and oxygen, and to remove dead neurons. The neurons are brain cells that communicate with other brain cells in this network to create biological responses in the organism. You see neurons everywhere around you. There seems to be no end to them. Your teacher tells you that one brain may have up to 100 billions of neurons. The diameter of a cell body or soma of a neuron is usually between 10 to 25 micrometers. A micrometer is 1,000th of a millimeter. As a comparison, a strand of hair has a diameter of 50 micrometers. Dendrites are protruding from the neuron cell body, like tree branches, and a cable-like extension, called the axon, extends for a millimeter or more. The longest axon in a human body is over 1.5 meters in human adults. The diameters of axons and dendrites are about 1 micrometer. On the dendrites, there are tiny mushroom-like receivers for the neurochemicals or neurotransmitters that are sent from other neurons across the synapse, the junction that separates one neuron from another. The synaptic gap between the neurons is tiny and barely noticeable. It's only 0.02 micrometers wide, or 2% the diameter of a dendrite. When neurotransmitters change the electrical chemical balance in a neuron, there is a nerve impulse or action potential that travels down the axon before branching off into several end branches that are called axon terminals. Axon terminals hold neurotransmitters in cavities called synaptic vesicles. If they receive an action potential, those neurotransmitters will be released into the synapse, acting on other neurons. You notice that the system of neurons is very complicated, as one neuron alone may be connected with a multitude of other neurons through the axon terminals. Millions of neurons may interconnect into neural networks or circuits. As you travel around the brain, you notice that some neural networks have more activity and neural connections than others, whereas others seem to communicate less. Some neurons even seem separated from others. Your teacher explains that this has happened through the process of synaptic or neural pruning. The more a neural network is used, the more effective the communication between neurons will be. Contrastingly, if a neural network is not being used, connections between neurons will fade. The process is fittingly called synaptic or neural pruning. The axon terminals and dendrites look a bit like tree branches, and it's necessary to prune some of them to develop a healthy brain, much in the way that pruning a garden will lead to a healthy growth of plants and trees. It's believed that neural pruning removes damaged brain neurons that are not in use to improve the network capacity of neurons that are being used. Neural pruning occurs frequently during brain development, especially in adolescence. It is kind of getting rid of the garbage that you don't use to have more space for the things that you do use. The general term for the making and breaking of neural networks through neural pruning is called neuroplasticity. Neuroplasticity is the ability of the brain or neural networks to change over time, either from experience or genetic factors. Neuroplasticity has a large body of evidence to support its existence. The process exists in all animals that are capable of learning. There are also many applications for knowledge of neuroplasticity, as it can be used for rehabilitation after brain injury. As it is the biological basis of learning, It may even have applications in education. Not all neural networks develop from experience, however. Teacher tells you that the development of some neural networks may also be genetically programmed, which may explain localization of function for many behaviors. Neural networks in the brain are rewired through neuroplasticity, but also through the creation of neurons, so-called neurogenesis. Neurogenesis is well accounted for in childhood, and in the hippocampus and olfactory bulb for adults. Some research suggests that the generation of neurons in adults may occur in other parts of the cortex as well, but the jury is still out on this one. Your field trip is now over and your teacher is bringing back the class to base. You have been truly amazed of glimpsing into what humans consider to be the most complicated object in the universe.